We start. Thank you very much, Dr. Aline, that you are here with us. It's a pleasure. I'm honored to meet you.
Thank you. It's my honor to be here. I'm super glad to be able to show to everybody what we are doing in the ISS.
That's great. I mean, we are keen to work with that. But Dr. Aline, I mean, let's start with introducing you to the Greek audience, to the Greek scientific community. Okay, Dr. Aline Martins is the executive director of ISCOR. And ISCOR means Integrated Space Stem Cell Orbital Research Center at the UC San Diego. Is that right? Did I pronounce it well? Okay, that's great.
Yeah.
All right. So, Aline, we followed you the last 3 days to one of our amazing trips. I think you felt that, right? Could you please tell us a few words what we have seen so far? What is the expedition and what we are doing? And where are you? I mean, amazing background.
We are sending experiments with brain organoids to space, to the International Space Station. And why we are doing that? Because we are leveraging space to understand and to try to find treatments for earthling diseases. So this is really not about curing astronauts as a primary aim. It's for us on Earth, is to accelerate the biology for us to understand better especially neurodegenerative diseases as Alzheimer, for example.
But before proceed with explanation of brain organoids and all those stuff and that we are doing so many years, how do you feel that you are close to a very great research project? You have seen the Falcon, the lift off. How do you feel? What is the? Could you please give us the vibe over there?
We are mostly certain tired, yeah, because as you can imagine, this mission begins 6 months before all the preparation. So we are a little bit tired, but we're still heading for phase 2, which is the integration of the ground experiment at the West Coast. So tomorrow I'm heading there back to the San Diego lab center to integrate the ground experiment of this endeavor. So, but I'm super happy to have this opportunity. It's a unique opportunity for any scientist, and at the same time full of hope, because this is why we are going to space, because we hope that the space can unveil key elements that we don't understand on the experiments on Earth.
That's right. So before telling us more, could you please tell us a few words about you, your career path, your research, what you are doing actually? Yeah, we need to meet you.
So I was today is being 1 year ago. Actually, I'm doing anniversary at the UCSD. Okay. So 1 year ago, I was hired. I was recruited from Scripps Research. But I am a Brazilian by my career. I did in Brazil. I am a Brazilian scientist.
All right.
But you can imagine, as a Brazilian scientist, I would never had this chance to go to NASA and to space. So to kind of leverage my career and I always wish to do like a better science, a bigger science that I was doing, because I was built to do this amazing thing. So so I decided to move to United States, and I think that was oops. It was an excellent decision. So it was an excellent decision, because as now I'm doing this amazing kind of research that everybody is everybody is now watching.
Okay, yeah. All right, so that's great. I mean, and your research is about what? I mean, the main research area.
So in this particular flight, we are flying 3 kinds of projects. We are flying a project that is going to understand what the space environment causes in Alzheimer. and we will test some drugs to revert the aging that the species causes in these brain organoids. The second experiment is one experiment about how complex construction of organoids that we call assembloids regulates the neuroinflammation on the brain. And the third project is a project about aging. We will understand how the brain organoids age in the space when they have different ages, as they are like babies or teenagers or adults. So we have different stages of age, and we are testing how the space environment will accelerate this biology.
Okay, that's great. So could you please tell us, what are brain organoids? What is the meaning of the real meaning of that?
That's a good question, isn't it? Yeah, because we have been talking about brain organoid, but what is brain organoid?
What are they? Yeah.
So brain organoids are mini brains. They are like avatars of ourselves. So how we do construct brain organoids? A lot of people ask me, are you going to biopsy the brain of people? And I said, no, that's not about that. So how do we construct the brain organoids? We get the skin from the patients and transform this biopsy into stem cells. From that stem cells, we guide the cells to become neural cells. And this is how we construct the brain organoid. So it's a well-known technology in medicine, but that Alison Watry and his lab especially developed to guide to construct the neural cells, okay? So you become from something that is not specific to something that is highly specific. So this is how we build this 3D culture.
And those organoids are living, are alive, or and how do you maintain this? The whole thing. How do you maintain to be as a living?
Absolutely, they are alive. Come on, they are our organauts. Okay, they are traveling. They are alive.
Of course. But how do you maintain this? How do you say that? This life for them. How do you maintain that in a bioreactor or whatever?
So the way that we are now sending this experiments to the space station is using autonomous cube lab. And this we partner with a company called Space Tango. Space Tango developed this big incubator that maintain the brain organoids alive when they are there. So it's a huge incubator that maintains them happy and healthy for us to retrieve the biology when they come back to Earth. So when they come back to Earth is when we begin to do the analytical experiments on them. So the analytical experiments are not done in the ISS because there is no support for this. Imagine to have a mass spec, gigantic mass spec over there. It's not possible. So we need to retrieve the biology alive to understand what happened in the space.
Okay, so I'm trying to learn from you. I'm trying to absorb the whole knowledge, but in reality, what makes them so powerful, the brain organoids, in order to understand how Alzheimer or autism or dementia affecting us? What makes them so powerful?
George, this is an excellent question, because if we remember, the brain is the only organ that you cannot biopsy. So you cannot really understand what's going on inside the human brain. So because of this, you need to construct models, especially disease models, okay? So the brain organoids are one of the best disease models that we have nowadays to study neurodevelopmental diseases as autism, as Rett syndrome, as well as neurodegenerative diseases as Alzheimer and Parkinson.
Okay. And okay, I got this. And what is the role of the space? I mean, we know the space is often seen as distant and futuristic, and the whole great hype thing right now with NASA and SpaceX. Okay, but Aline, you are leveraging this ecosystem of space. I mean, you are.
So this is our ninth flight. Okay, so we learn a lot with all the flights. So in the previous flight, we didn't know what was going on, but when we retrieved the biology, we find out that the organoids were aging faster. And not just this, they were aging in a special way, a phenomena that we call SINS, which is SINS, SINS. That means space-induced neurosenescence. Okay. So they were aging in a unique way that could be only done in space. And we use this to create older brain organoids to mimic neurodegenerative diseases. So this is why we use space to accelerate the biology.
Okay. And by those results, again, to your lab with a mass spectrometry, you are capturing what? The paths that they are doing the whole thing. So in order to break the paths and understand better how dementia is affecting a person, is that correct? Or
so the mass spectrometry today is so important that the first findings that we discovered was because of proteomics, mass spectrometry based proteomics. So with these findings, we discover how the brain organoids age, that the brain organoids do not all the cells age in the same way. So there are cells that are more affected with a space organoid. So this is how we apply analytical technology, cutting edge analytical technology as mass spectrometry in a very good disease modeling as brain organoids to study neurodisease. So this is why we need to retrieve the biology alive. Okay.
Of course.
And this is how we find out all of these incredible things that were happening in the space. So with these clues now, for this experiment, we want to test drugs, test therapies for Alzheimer, for example. So because of the findings that we acquire in the previous flights, now we can go further. We can chase therapeutics for this kind of diseases.
Okay, that's amazing. That's very hopeful, and that's brilliant work that you are doing. So by that, and I want to emphasize that because we discussed this before, by the meaning that you are testing drugs, so that means that you are not, let's say, discovering a new drug, but you are discovering a new way to use an already drug in the market. Is that correct?
That's exactly. We are not testing new molecules. We are discovering new ways to test, to have another approach to test these drugs, especially neurodegenerative drugs. Because imagine that to test something for Alzheimer, you have to cultivate the organoid for 1-2 years in the lab. So it's a long time for us to test it. Yeah. So we are using space to accelerate the biology and to shorten the pipeline of discovery.
And by example, I mean, on this expedition, you are sending for 6 months in the space right now, right?
Yes, because this is a very different flight.
And by setting them in 6 months in orbit, as an example, as a rule of thumb, what is let's say the gain of the human time that you may gain on that? I mean, you don't know that, huh?
That's another excellent question. I mean, the other director of the center, that Alison Waters, says they will come back with 800 years old. And I said, Alison, we don't know that. But so that is a question for itself, you know. We don't know how much, we know how much they age for 30 days, 40 days, but not 4 months, 6 months. Yeah. So this is uncharted waters. We don't know really how, if it was going to be linear, if they will stabilize. We really don't know. We will find out.
Okay, that's great. And what are the, you know, right now, the biggest scientific barriers that you are currently facing in brain organoids? What are the barriers that you are really challenge you and your team?
What are the what?
The biggest barriers, the biggest challenges of the brain organoids, the scientific ones. Yeah.
This is amazing also, because what we need right now, it's because to send the brain organoids to space is still a little bit expensive. It was much more expensive. Now it's much more cheaper because, for example, SpaceX reutilized their rockets, so it's much more cheaper to send now. But it's still a little expensive here if you compare to the ground. So what we need is to find other ways to fund these experiments. We need to go more there, because with 1, 2, 3 flights, it's not enough to establish for sure that phenomena happens.
So you need more data, more samples, more, you know.
Yes.
Okay.
And also, there is a lot of improvement in the Cube Lab itself. I mean, a lot of other, not just Space Tango, but other companies are trying to improve the Cube Labs to test the drugs up there, which would be ideal.
That's great. Okay, so one thing is that you need, of course, more data, more samples in order to feed your model, right? I understand that. The other thing is you need more, let's say, advanced technologies. Allow me to say, I mean, in order to be as a very technologically cube, in order to maintain the brain organs in life. Another, is there any analytical challenge right now or not?
I mean, we are all over the place. Yeah, we are trying to improve the box. We are trying to embed new technologies. We are even trying to build our own Cube Lab, you know, with our own ideas. But it's still, the reality is still a little bit further. So we're still chasing the better ways to do that. The reality is that for now, this is the only way that we have to test the biology up there. So we are all over the place talking to companies, biotechs, to improve all the things that we can do in the space.
Okay, but in the lab, is there any scientific challenge for those results? I mean, is there anything that really keeps you awake on the night?
Everything keeps me awake at night. Come on, George. Not being fair. I'm thinking about science all the time, you know? So I think that's not a worry, but a challenge. As you know, in science, when you ask a question, 10 questions pop up. So the questions that are emerging or rising from these experiments are numerous. So this is what keeps me awake, but at the same time happy, because it's an endless well of knowledge. So we still have to learn a lot, but the things that we are learning right now are amazing me, not just me, but the whole community. So not the limitation per se, but there is, of course, always a lot of work, especially when there is a splashdown. But everybody is working happily because everybody is curious to understand what's going on over there.
One more question about the brain organoids. Is there any limitation on the diseases that you can cure? I mean, is only brain, or from the stem cells that you are, let's say, sampling from a patient, like, you know, you can also guide them to be whatever. So might be cure other diseases as well, not only brain, like Alzheimer or dementia.
This method to transform skin to stem cells actually was when won a Nobel. It was the Yamanaka factors, okay? So they are really well known. So you can guide to transform a stem cell in whatever you want. We use brain organoids because we work with neuroscience. This is a background from Alison Watry, and we embrace it. But there are other people that are working with cardiomyocytes, liver, intestine, kidney. So you can guide to whatever is your area of expertise. But we work with neuroscience.
Okay, and as far as I understand, about this and then the future, again, space missions that we are going to, of course, execute, is that the first win, and I'm looking positive on that, the first win will be for the humanity here in Earth. It's not only for the space astronauts or the space. I mean, we have to differentiate this. I mean, it's of course win-win because the space astronauts, they may get stem cells by themselves, so they can cure whatever. But this win for the neuroscience is here for the Earth, right?
I didn't quite understand the question.
The question is that once you are taking all the results and you have really positive results that this drug, this market drug that cure dementia or Alzheimer, the first win is here for the Earth, for the humanity. It's not for the space itself, right?
Yes. I mean, the first aim is to help society on Earth. Of course that the astronauts will be beneficiaries of these discoveries, but our center is working towards new therapies to earthling diseases. Is that your question?
Yeah. Of course. And
so of course that the astronauts will be beneficiary from that. Our aim to colonize will beneficiary from there. We are actually aiming really the earthling diseases.
That's great. So Aline, one last question from my side, and this is quite personal. So if you look, let's say 10 or 20 years ahead, what's the most hopeful vision that you have for this field?
I will tell you some things that I learned during this full year working with space. Space is the new Old West, okay? So space is the new economy, okay? And there is a very prolific environment that is going around all these technologies. So I believe that since we are hominids, first humans in Earth, we look to the sky. Now we are in the sky, and I don't think that we will come back ever from there.
Okay, that's great. So Aline, thank you very much, and thank you very much for giving us also the opportunity for this live blog. I mean, we have actually thousands of Greek citizens that follow the live blog. This is the stats that for the last 3 days. Thank you very much that you shared all those insightful new research projects that you are working with the research. We wish you congratulations, and we wish you the best luck for your results.
George, it was a pleasure to be here, and you will be with me on phase 2, okay, which is in the West Coast. I will send you pictures and try to do small movies about it, and maybe we can recap all of these 2 phases together in the end of the week.
Of course, that's great. Thank you very much, Aline. Thank you very much.
Thank you, George. See you.
Best luck. See you.
Bye, everybody.
Bye.